Dr. Daniel is our soil and water conservation specialist, and he's leading this session. You want this or the lapel?
Yes, please.
Okay.
Thank you, Brian. Have you guys ever looked at, if you look some of us up online, it has our like experts profile there, and there's a nice blurb about us. We write those ourselves. So that would be as cringy as like me sitting up here introducing myself with all those things, which happened to me at a talk. Somebody read all that out in front of me while I was standing here, and it sounded worse than I thought it did. So I will spare you the horror of that experience and just say, I am Daniel. I am the soil and water conservation and management specialist here. So we'll go into another session for Soil and Water Con. So thanks for Dr. Sharma giving us some good water-related conservation stuff yesterday. So this session will focus more on soil conservation. So with that, there's always opportunity to be thinking about new things, and especially, us folks in academia, we spend a lot of time thinking about very specific things. XYZ. I am guilty of not knowing my own discipline as well as I should. So in a little bit of selfishness, I wanted to dive into this topic and discuss kind of what are we really looking at with soil organic matter. I mean, it's still, I find it very interesting that soil health and regen ag continue to be very popular. They continue to be financially supported by a lot of different entities. It's something a lot of us inherently believe in, but the problem is it can be hard to connect data to that sometimes. So I just want to touch on some of those thoughts today really quick. Okay, I have never been here. Has anybody ever heard of this or seen this facility? It's north of Tucson, Arizona, apparently, and it's called Biosphere 2. It's a mega greenhouse complex facility. Dr. Ferris is nodding her head. Have you seen it before? Have you been in and explored? Okay, this was apparently a very interesting biological and social experiment. Originally, the idea was this would be an enclosed ecosystem, and they threw a few scientists in there to live and try to survive off their own food that they were growing. They gave them a few pigs. There were some goats. There were some chickens, and there were different areas apparently of the biosphere where they had like a rainforest. They had a mini ocean and a coral reef and a desert ecosystem, and then an ecosystem they were farming and trying to grow their own food. As you would imagine, the experiment didn't last very long. To their credit, I think they lasted about 2 years, like living in this space and solely eating what they could grow. They made a couple massive mistakes. One was the idea that a few people could get along in an entrapped space for a while. So apparently there were some major factions and politics going on that they had issues with. The other massive ecological mistake they made was they brought in, for the agricultural soil, a pretty good soil with pretty high organic matter. And I think all you guys know where I might be going with this. What happens when you disturb soil? Where does all that organic matter go? CO2, right? So they were having these huge inputs of CO2 into the system. It was enclosed, like the atmosphere was enclosed in here. What happens when microorganisms are decomposing organic matter, right? They're using up oxygen. So the system started dropping in oxygen at an alarming rate. So it went from roughly atmospheric 20% oxygen, apparently down to like 14%, which is like living at 14,000 feet elevation or something like that. So they had to can the project. I think Ashley is it more like a visitor center kind of thing now today. So still very cool. And I admire their idea. But I bring that up to say, we all know that soils are very dynamic, right? And especially this idea of soil health and building soil organic matter and building soil carbon and what that means, right? It's not a steady process. And thankfully, it's not a steady process, or we would be living in mounds of poop and debris all the time, right? There are things utilizing that resource. There's energy in organic matter, and soil microorganisms are utilizing that, and it's helping cycle carbon through our world, right? But there are different components to that, obviously, like inputs are important, the type of input, Where those inputs are being placed in the soil is very important too, in determining what can be utilizing that resource XYZ. A portion of that will be chemically oxidized for energy, right? Microbes are doing biologically the same process I'm doing right now, right? They're releasing chemical energy by respiration and blowing out a lot of hot wind when they give presentations to each other in the form of CO2. You can be exporting organic matter, right? In crops, or exporting organic matter, any residue management, XYZ. Very important to Oklahoma, obviously, is erosion. Sometimes we don't think about, there's a lot of organic matter in the soil water that's dissolved. It's in that solution. So you can leach out soil organic matter as well. So kind of the philosophic part of the presentation that's interesting is, on one hand, soils are very dynamic and the carbon cycle is extremely dynamic. But on the other hand, there are parts of the carbon cycle that are extremely slow. So you have these altering systems kind of going on within a soil itself, right? We know, kind of working on carbon dating or isotope work, there are portions of soil organic matter that are easily thousands of years old, can be more than 20,000 years old. So the turnover time in certain parts of the soil can be extremely slow. So there's portions of soil organic matter that never change, basically. So that's kind of it has stumped us as soil scientists for a long time. And to be fair, it kind of is still stumping us, but I think we're getting a little close to understanding. But the traditional reason that we thought this was happening was, and so we'll all go back to intro soil science class, right? We've all done this. We know what determines these carbon cycle within a soil, right? Temperature is super important. How much water there is. The microbes need the right soil moisture to be efficient. Obviously, are we putting more carbon into the system? Are we taking carbon out of the system? Are we tilling XYZ? What kind of microbes do we have there? But the long-term school of thought, and you guys remember the fancy word, right? Recalcitrance means something is hard to decompose. So if it's hard to decompose, it's not going to go away. And the idea was that, so quality and structure of organic materials in the soil is what was really driving how long something stayed and how quickly it turned over. So high quality materials, we expected the microorganisms to use those very quickly, decompose them very quickly, right? But maybe some other things like woody materials, lignin, things below carbon to nitrogen ratio, they had a higher chance that maybe they would hang around longer. So that's what we thought. That's what I learned as well in my soil science classes. Okay. Here's how I see soil science. And there's a lot of soil scientists in the room. I'll be curious if you think this is a terrible analogy or a good analogy. There are 6 pictures behind this black wall, okay? In soil science, we're trying to learn something about an individual soil from an individual field, right? And I feel like every analysis we do is like just poking a hole through this black wall. We can see a tiny little piece of the puzzle behind that hole we poke through the black wall, but it's hard to put them all together. So, for example, we get in the habit of just jabbing at the soil in the same spot all the time by doing the same test in the same way to every soil, right? And by doing so, we assume that maybe we can tell something about what's behind the curtain. So just for example, let's say if we're using pictures as an example, we did a test that revealed the small portion of the picture in the top left corner. Right. So in a soil, that might tell us something about the organic matter. It might tell us about the nutrient status, right? Well, in this picture, maybe it tells us something about the sky. Okay, so there's a few pictures there that look identical. Blue sky day, right? Some of them are cloudy. Maybe that tells you something about the season in the picture, right? Maybe it's winter, maybe it's summer. We don't really know. Same with soils, right? We do one nutrient analysis. pH is very important. But without putting it into context of everything else, it can be hard to interpret exactly what that means in a soil. Okay, so let's do another test. Let's probe at the bottom left-hand corner of this picture for each of these 6. Okay, now we see something about the vegetation, which is interesting, right? Maybe that's grass, maybe it's wheat. I'm a little more confident that that's some sort of range or pasture because I see more bunch grasses, right? That I'm seeing some bare soil. You get the picture, right? We have the same thing with soil samples. Some soil samples, based on their values, reveal more than others, even within a test that we're doing. Finally, let's probe kind of in the middle of the picture. Now things are starting to come together, but the problem is no matter how far you go, no matter how many tests you run, no matter how many soil health indicators you run, it can still be hard to really understand a lot about that soil and a lot about that ecosystem out of context, right? So anybody want to take a stab? Are any of these pictures not from Oklahoma, I guess would be one question I'd ask you. These are just pictures on my phone. These are lupins. I don't know, honestly, if we have those in Oklahoma. I've only been here a year. They're pretty, though, right? So it tells us something about that. That looks like corn. Could be a lot of different places, right? I see kind of a hill or a ridge up there. Well, here's the whole picture, right? Again, it can just be hard to tell what you're seeing based on kind of soil samples we do. So the soil scientists are doing their best. A lot of times we bias towards doing as many different tests as we can to try to see the whole picture, or maybe for constraints we just try to do samples that reveal the most information. Like if we could take a sample right on the horizon of each of these pictures, it might reveal more information than one that just takes a snapshot of the sky, right? So that's me getting a little philosophical, but I'm trying to prove a point today. Okay, so we go back a couple hundred years. Attempt one at this process was a guy named Franz Achard. He was literally kind of a mad scientist, I guess. And these guys, your taxpayer dollars are hopefully having us do some good research. For whatever reason, this guy had a job and he could do whatever he wanted. He liked tasting things, apparently, as part of his chemical analyses. He's most famous for increasing the efficiency of sugar extraction from beets. So I come from sugar beet country, so I appreciate him. He got really interested in soils and heavily organic soils, and he started trying to understand what was soil organic matter. And humus is a word that comes from Latin meaning low, right? Like humble, or same root, right? So what he did is he did something actually quite profound, is he just put a lot of base on a soil. And he found that if he raised the pH very high, he was able to separate out the soil organic matter. So some of it was soluble and some of it was not. So then he took what was soluble and he changed the pH again and he made it very low. And again, some of it reprecipitates and some of it remains soluble. This is the model we have used for a couple hundred years. And this is the humic or humus model, right? And a lot of you guys probably learned about this. And the idea was there are things in the soil that we can extract at different pHs, but they are very long lived. But what he found was actually pretty profound for his time. And here's why it works. So what he basically inadvertently discovered is there are a lot of pH-dependent functional groups, very complex, different forms of soil organic matter, right? And they change across the soil organic matter structure. So if you, for example, just have something acidic, so this is a carboxyl group, back to intro biology, right? If this is in an acidic environment, this little proton has no desire to dissociate, right? So this carboxyl has no charge, but if you put it in high pH, this will dissociate to produce acid, and then this little active zone develops a charge, right? So things solubilize when they're charged because they associate with the polar water molecule better, and they nice snugly fit inside water and dissolve. So that's what the humic kind of theory, the humus theory, was doing, was saying, okay, there's something going on in the soil where we are decomposing a lot of stuff. Some of it might be hard to decompose, some of it might be easy to decompose, but as the microorganisms are working on it, they are changing the structure of it. They're making it incredibly diverse and very complex. They are giving it a lot of different functional groups, and that's what really helps it stick to the soil, and that's what really helps it persist for a long period of time. So that was the school of thought. So we broke down soil organic matter into different compartments, right? We had plant litter, or even microbial litter, right? We had dissolved organic matter or dissolved carbon that's just kind of floating around in the soil water. And then we had this long-term stuff that doesn't turn over very well. It's more stable and it's harder to change. On the other hand, it's harder to build, right? So that was the humic stuff, substances like human humic acids and fulvic acids, right? So again, this is a classic soil science figure, right? And the idea was management was influencing soil organic matter or soil carbon as a portion of soil organic matter. And any disturbance was causing more microbial activity, and we were losing large proportions of organic matter after we start disturbance, right? But the proportion of organic matter in different pools was not equal, right? So we lose things that are theoretically easier to decompose first. So we would lose plant material and residue. Then we start losing these active pools of soil organic matter, which we call labile carbon, if you've ever heard that word. It's just a silly soil science word. That means it turns over relatively quickly. So it was more sensitive, right? Harder to lose that humus or that stable organic matter. On the other hand, when you revert back to a system that's bringing more carbon into the soil, it's easier to rebuild the short cycling stuff, and it was harder and slower to rebuild the stable stuff, right? Because we're assuming it takes a long time for the microbes to do all their work and stabilize that stuff. So I'm sure I'm preaching to the choir. I feel like you guys all learned that in soil science class at some point. Again, I bring that all up to say, for a long time, we have been stuck doing something that is not a whole lot more profound than tasting the soil and deciding if it's bitter. Because really our arsenal of things, tools we use to try and shoot through that picture and see what's behind the soil curtain is still pretty limited, but they are based off of this idea that we need to measure different portions of the soil organic matter pool to determine if something is cycling quickly, something is slow or stable, and things like that, right? The problem is that we continue to have is that the links between soil organic matter and soil function, even though we all kind of inherently believe in them, which is true, they're rarely directly demonstrated just because it's so hard to prove causality, because there's so many factors influencing how a crop grows in a field, right? Even though we know soil organic matter is incredibly important, it's hard to connect that. So here's some things you may see. Different labs, commercial labs are starting to offer this stuff. I don't know if any of you folks have customers or clients who are interested in like a soil health type test, but we'll break down what's in those different available tests. So we all know total organic matter or total carbon. If you took a tour of Swaffle yesterday, this is really dependable. We just basically burn the soil really hot and we see how much CO2 comes off. Total soil organic matter is just weight change based off of burning a sample as well, so we're not directly measuring CO2. But what we're getting is we're getting a look at the whole soil organic matter in the soil. It does not determine or distinguish between something that is actively cycling very fast or passively cycling, right? And we have problems with that. Are we picking up roots? Are we picking up plant residue? Are we actually picking up humus, et cetera, things like that? It's pretty easy. It's pretty affordable. The problem is it's hard to pick up a change when you invest in new management, right? 3 years of moving into no-tiller cover crops, it can be very hard to actually detect if we're building soil organic matter, because you have to take so many samples to overcome the high variability in soils, right? So people got interested in looking at different types of soil organic matter. So they wanted to understand if there were different portions of carbon or organic matter in the soil that were more sensitive to change. So there's different things they looked at. They looked at, you've maybe seen this, water extractable organic carbon. So it's carbon that we can pick up from a soil sample by just shaking that soil sample in water. So that carbon comes out into the sample. We can analyze that sample and see what's in it. It's easy. It's theoretically more sensitive to management because the amount of carbon kind of available to microbes is dependent, again, on those more quick cycling pools. So this picks up on it. Some problems are it changes a lot seasonally, so it's hard to see the signal through time. There have been arguments about standardizing that method, and it's actually can be hard to reproduce that data, so it's highly variable. Some of you guys may have heard a funny word called POXC, which means permanganate oxidizable carbon. It's just applying a special chemical to the soil that will oxidize some of that organic matter that's easy to get to in the soil. So it's considered some of the more active organic matter in the soil. It oxidizes it or reacts with it very quickly, and we can read the result of that based on a color analysis. So it's easy in a lab. It has been shown to be really sensitive to management. It gives you some idea about the quality of organic matter we're dealing with, but it deals with some not so nice chemicals, so that's a disadvantage of that one. A pretty popular one that we'll talk about at the end is called particulate organic matter. This is measuring active forms of organic matter in the soil. So again, it's been shown to be more sensitive to management. It's relatively well defined, but again, there's some different ideas about how to get at and break up this particulate organic matter because it's a relatively arbitrary term. So then we move into the world where we want to actually understand how microorganisms are using soil organic matter. So you guys might see things called sea mineralization tests, CO2 burst tests, things like that. Basically, we put soil in a jar, we let the microorganisms start decomposing that soil, and then we measure how much CO2 they produce. So that gives us an idea about some of the easily accessible carbon or organic matter in the soil for microbes to decompose. So that's nice. It also tells us how easily they can use it. So not only does it tell us how much there is, but it tells us what maybe if it's good quality organic matter for them to decompose, and then how those microorganisms are actually functioning with it, right? The problem is there's a lot of different methods and it's relatively slow. Some of them take up to 28 days. Okay. Microbial biomass carbon, we're interested. So we obviously know those microbes are chewing on that carbon. They will respire a lot of it away as CO2. They will take up a lot of that into their tissue, right? And that's what we call immobilization. If you have poor quality straw or something on the field, right? Those microorganisms can be robbing nitrogen and nutrients from the soil because they need to actually take up some of that nutrition into their microbial tissue as their populations grow and expand while they're decomposing stuff, right? So we like to look at how big the microbial population is, and we can do that with fumigating the soil and measuring how much microbial residue comes off. PLFA, you guys might have heard of that too. It's called phospholipid fatty acid analysis. That tells you something about what types of microorganisms are in the soil. It gets you down to the functional group level. So kind of their PLFAs that they're leaving behind have different structure, and you can tell if it came from like a certain type of bacteria or a certain type of fungi, like an arbuscular mycorrhizal fungi or something like that, right? It's slow, it's expensive, and you use chloroform. And I accidentally chloroformed myself a little bit once doing that, but that's a story for to share over a beer and not right here. And finally, you've probably heard about soil enzymes. It's a test we can do where we basically can get an idea of how many or what types of enzymes the microorganisms are putting out into the soil in order to do something. So they're using those enzymes to break down certain types of organic matter, certain types of that carbon chain, right? Cycling nitrogen, cycling phosphorus, right? Dr. Dang is a world expert in this. And if you saw her student Grace's poster yesterday, they're super sharp on this. So we have a lot of expertise in OSU at that. All this to say, so, you know, each one of these would be a picture in that puzzle piece. If we could do all of them, it may tell us quite a lot about the soil. On the other hand, the picture can still be very fuzzy, trying to see what's behind the curtain. I bring all this up because it affects some of these tests that are getting popular. So not supporting any particular lab, but a quick AI or Google search will give you commercial labs that are doing this stuff, right? So not only can you do soil health tests, like the Haney test you guys have probably heard about, you can do enzymes, you can do PLFAs, you can do POXC should your heart so desire that, right? And my heart does desire that. I love that. But it's expensive, right? So it depends on if somebody has interest. Oh, we can determine if you have great soil health. All right, let's send this sample off and see what happens. Different lab, Haney test, enzymes, pretty expensive, right? But nice. Here's another one for nitrogen, certain type of nitrogen. Another commercial lab, soil health complete analysis, includes a Haney test, includes all of your nutrient extraction, XYZ. We get the picture. Okay, so there's really two, to my knowledge, two soil health tests that are routinely used among folks like us, not just doing research, but for more of a broad scale approach. And one is the Haney test. And I am not here to knock on any tests or really promote any tests. I just want to help you understand what they do and don't do. Okay. Is Dr. Arnell wanting, okay, the folks online couldn't hear anything, so we are safe. All right. The Haney test does a traditional NPK plus micros test, but it uses a special extractant that Dr. Haney, who was part of the ARS, he developed this in Temple, Texas for a long period of time. And his logic is good, honestly. So he thought his special extractant is one you would not get from Swaffle, but it mimics how plant roots are getting nutrients out of the soil. He's only using one extractant, so his idea was it's reducing lab costs and making throughput easier. So he gets stuff out of the soil by doing that. He does this CO2 burst, where he puts soil in a jar, he gets it wet, and he sees how much carbon it produces very fast. He used a fancy paddle system to measure the amount of carbon that came off. Something very important to this test is that water extractable organic matter as carbon and nitrogen. So basically you just shake that sample in water. How much of the organic matter goes into that water can be analyzed. And the idea is that that's the organic matter that the microbes would actually use in a season to create or release nitrogen, nutrients, etc. for your crop, right? He mushes that all together in an equation and it gives you a soil health score. I am bad at math, so I did this for me, not for you. If you have more CO2 production in a sample, it's going to increase the soil health score. If you have more extraction of nitrogen from the water in the sample, it's going to increase the health score. And this is not just nitrate and ammonium. This is also nitrogen in organic matter, especially nitrogen in organic matter here. If you have more carbon that comes out of the water, it's going to increase the test. But one thing that's important is if you have disproportionately too much carbon to nitrogen, so poor quality of that stuff you pull out, it's going to decrease the score you get, right? What I find more interesting in the Haney test is he took a stab at trying to figure out how much nitrogen your soil would produce in a year. And the basis of that is he can do it with phosphorus as well, but this is nitrogen here. He takes the value of the water extractable organic nitrogen, he multiplies it by the CO2 production. So what that's saying is he's trying to understand how much organic matter is decomposed in a certain period of time. If there's a certain proportion of that is nitrogen, then theoretically that's more nitrogen production that could be made available to your crop in the growing season. He multiplies that by a rainfall factor to try and get at some environmental component. So if you're expecting more rainfall during the year, he's expecting more nitrogen output. And then he adds the true plant available nitrate and ammonium to that. And he gives you a number. I have only seen a Haney test once. A hay grower in Wyoming gave it to me, which was crazy because those soils have like 30% organic matter. So the Haney test was off the charts, right? And they were predicting that they were going to like make hundreds of pounds of nitrogen available per acre every year, which is not true because those soils are cold. So there are flaws, right? But I appreciate the attempt. It's nice to try and get at how much nitrogen might actually be made available so we can change our nutrient recommendations. Plug your ears, Brian. I know it doesn't work that well. That's just the science of it. So there's been a lot of criticism, and rightly so. Frankly, a lot of times it doesn't show any differences in soil health. So the Haney health score is too generic. So here's different cover crop implementation. So no differences against the control for cereal rye wheat, rye plus legume cover crop treatments after several years. Can't pick it up in the Haney test. The big part of the Haney test is that there is good correlation between actual nitrogen that comes out of the soil and his CO2 burst method. Other researchers have found that the correlation is actually quite poor, so it's hard to predict. On the other hand, some folks have looked at using the Haney test nitrogen recommendation in corn in the Midwest, and it is related, frankly, very loosely related, so it's not all bad. But they were saying if the Haney test recommended a certain amount of nitrogen by crediting you some back, it was basically under predicting slightly how much nitrogen you would actually need with a tremendous amount of variation around that. But the idea is, right, we're all about end credits and we want to manage nitrogen efficiently. So we don't knock the concept completely, but no, this is soil science, right? We can do a test, but it's hard to see what's actually going on. It's very hard to predict what's happening in a soil. Again, this is interesting data. So this is the Haney test versus the optimum economic N rate. And you can see this regression looks almost exactly the same to this regression. It's because the Haney test really hinges on that CO2 production part of it. So that one test is pretty much driving the whole Haney score, basically. So one very small component of the test seems to be the strongest, most useful part of it. So just a take-home message there. Okay, there's another one. It's the Cornell Comprehensive Assessment of Soil Health, which is very cleverly called CASH. I hope to also create a clever acronym at some point in my career. But hopefully within the next 20 to 30 years, I can accomplish that. But for now, I'm running low on ideas. Anyway, CASH does maybe a better job, debatably, because not only does it take into account soil organic matter, but it takes into account more biological and physical properties of soil health, right? So it does an aggregate stability test. It does a penetration test. So it asks growers to do that themselves in the field. There's a soil hardness, kind of surface hardness thing going on. They get at different types of soil nitrogen. There's that POXC that I talked about pops up. It's active carbon. They also have a CO2 test, although it's different than Haney's. And then some soil physics like available water, right? So for each test, they put it into an expected distribution. So it's like, remember when, okay, my good friend's kid is very tall. He's a very tall baby. He's been like consistently in the 99th percentile, so we're hoping he will be a tremendous athlete someday for height. He's always like 99th percentile, right? Same thing we're doing here with soil. You can score a soil based on its value. You can say like, oh, it's in the 99th percentile of soils in that region for that value. So we'll give it a high score, right? So that's how that works. There's different thoughts about the different components. So there's the more is better theory. So like more organic matter is considered a good thing here. Less is better theory. So if you have less penetration resistance, that indicates a healthier soil. And then there's some that require an optimum range, right? So soil pH, they want it, closer to 7 and not on the ranges. So that's the idea of how they score it. Each one gets a score 0 to 100, and you get an average number, okay? The criticism of the CASH test is it was primarily done in the Northeast, mostly done in New York. They have tried to expand that. Again, there is just a limited ability to actually detect change. So this was like a tillage trial done in North Carolina. Over more than 20 years, they had different sites. This is only one. They did the whole CASH analysis on the stuff. Here's the individual components of the CASH and their score, 0 to 100, right? So they only actually found statistical differences among, so this is no-till for a long time. This is conventional till, and these are two conventional till organic systems. So they really only found differences in that POXC active carbon. Nothing else showed differences, and the overall score did not show any differences. Okay, so let's not do any soil health testing. Wait, same exact data set. They go back to it 2 years later. They analyze it with a different statistical method, and suddenly the world changes, and it's wonderful. They start picking up all these different statistical differences, and they're even able to do the impossible in soil science, and they're able to relate positive change in crop yield to several different soil health indicators that were done on that test. I say that because I think that says something important. Again, there's a lot of interpretation in soil health tests, right? There's still a lot of arbitrary human thought. We do not have a perfect way of scoring soil health. To be fair, I don't think we have a great way of scoring human health in some regards. I took my little health test. OSU offers 40% off your monthly premiums for your health if you go do a little health test every 6 months. So I did that, and I did pretty good. It was kind of like this, it gave me a lot of green bars. I was feeling good about myself. Apparently, my heart rate was 59 beats per minute because it was early in the morning and I was asleep while the nurse was doing my blood pressure. So I got a yellow score for my heart rate because it was too low, right? Whatever. Am I really concerned about that? On the other hand, am I healthy enough to run a marathon? No. Health is relative, right? So we have all these silly arbitrary cutoff lines for human health. We're struggling with the same things in soil health, right? So all that to say, people will continue to try and refine this through time. This is the new one. It's called SHAPE. Brian, do you know anything about SHAPE? Have they kind of dropped that? Yeah, so this was kind of a big paper 4 years ago, and I haven't heard anything about it since. But really, it's just an improvement of the Cornell test, and all they're doing is they are kind of grouping soils by what you would expect. So they're saying you don't expect as much organic matter in a hot, dry soil as you do in a cold, wet soil. Simple as that, right? So we will continue to work on this, but the picture will still be fuzzy. What is more interesting, and I want to just touch on it briefly, is there has been kind of a monumental shift in how we look at soil organic matter in agriculture over the last 10 to 15 years. And here was that idea about the humus theory, right? We put that soil in acid or base, and then we get out these really complex structures that we call humus. What is emerging is that the extraction itself is causing the construction of those very complex soil organic matter materials. So we think they don't actually exist in the soil. What is actually probably happening is soil organic matter is being built by just the fact that stuff gets trapped in different small parts of the soil, which is very simple but very profound, right? So if you get organic matter that gets stuck between two clay platelets, it's hard for a microorganism to use it and decompose it later, right? We are building bigger aggregates. Bigger aggregates may not hold that much water in them, right? So again, it's just microbial accessibility. Sometimes they may be able to get at it, sometimes they may not, okay? That's the new understanding. And the big driver of this is the rhizosphere, right? We know a huge majority of the microorganisms are living and active in that zone that is really close to a root. So as those microorganisms are chewing on organic matter, they are not particularly efficient. Some of it just goes into places of the soil where they will never use it again. So at 11:30 last night, I asked AI to create a picture of a small child, roughly 3 years of age, in a carpeted living room with a TV in the background. You have to prompt it correctly, okay? And I was very impressed with what it gave. Sprinkling glitter throughout the house joyously. And this is what it gave me, okay? So in a way, I picture the microbes doing the same thing with their little food sources that come off a plant root, right? They are joyously utilizing a resource. In doing so, they are not very efficient all the time. They're sprinkling stuff all out the soil. It will be impossible for mom to vacuum all the glitter out of that carpet. In the same way, it is impossible for the microbes in future generations to find all that organic matter in different parts of the soil. And it can actually be something that would be easy to decompose. It could actually be something a microbe could use as food very simply, like an amino acid. But if it's in the wrong place, they just will never find it and they will use it. What does that mean about soil organic matter management? It means there's a capacity that we can hit. There's only so much soil organic matter we can put in a soil because there's only so many places to hide it. But basically, we are finding this is a pretty sensitive indicator. So Dr. Yilling used to be here. She does a lot of work on soil particulate organic matter and mineral associated organic matter, so trapped organic matter. They found that moving to conservation tillage increased these trapped organic matter pools. After just 3 years, they could pick up on it. And cover crops did the same thing, which I think is interesting. So take homes, soil analysis is fuzzy at best. We're still in that world. So use a soil health test with a grain of salt. It's not all bad. The thing you can do with a soil health test is always compare apples to apples, right? Because there's so much variation. And Ala Estrada, one of our students, is doing this. If you saw her poster yesterday, she found huge amount of variation in the cash test in just one of our plot studies here at OSU. And it was just based on space. It was based less on treatment, right? So compare apples to apples. If you're changing something in your system, that's when a soil health test can be most valuable to see if you're picking up change, right? Focus on management-based changes instead of quantitative changes, right? If you want to see if something is improving soil health through time, you may be able to pick it up after you make a change. So those sensitive indicators can be useful in helping gauge the direction of if something is maybe moving in a positive direction or a negative direction. And then I think in the future, we are in kind of an exciting area where our new understanding of soil organic matter formation and how we're learning more about microorganisms will help us kind of tease these things out better. So rethink this picture is correct, but the driver these days that we understand is just if you have a soil that has good structure, that's building structure, has less disturbance, that's what gives us the better chance of increasing organic matter. So I will leave it at that. I don't know how I did on time, Josh. Oh, that was perfect. Okay. One question. Okay, we'll get on to better things. This is Dr. Hannah Rogers. Hannah and I did our PhDs together at Wyoming. Hannah grew up in St. Louis. She is one of those people I really appreciate who has a beautiful perspective on agriculture because she's seen it in a lot of different places of the world. She worked in Nepal for a year. Fun fact, she is semi-fluent in Nepali. For her PhD work, she did a lot of work on Kern's intermediate wheatgrass, so she's going to talk about that today. Currently, she's with the University of Wyoming. She's an extension specialist for soil climate resilience. So with that, thank you for being here, Hannah.
Thanks, Danny. I'm going to see if I can get this thing working. Okay. Can you guys hear me okay? Is this picking me up? No. Let's see. It's good? Okay, great. All right, thanks. I'm so glad I get to go after Danny. That was so good. And it really, I feel like, preps us to think about a lot of the things that I'm going to be talking about. I'm talking about this new experimental crop called Kernza that was developed around a lot of these soil health ideas that Danny was talking about. We're going to be jumping around a little bit. Hopefully it all flows, but I want to give a lot of background about Kernza, what it is, why people came up with this idea, and then dive a little bit into the Kernza work that I did during my graduate work, some of which with Danny. And then move on to some conclusions and hopefully some time for questions. All right, so Kernza came out of this idea of natural systems agriculture, which is the idea that we should farm in a way that mimics the native ecosystem. Well, how can we do that? So this is a rainforest system. It provides a lot of these great ecosystem services like water cycling, nutrient cycling, habitat for wildlife. And when we convert it to monoculture systems, we lose some of those services we care about, right? We hear a lot about this in the news about the rainforest. So the idea behind natural systems agriculture is we still need to farm. We need to make food and fiber. So can we farm in a way like this farm? This is a natural systems farm where we're growing many different types of crops, many different levels of crops, many different groups of crops. There's some coconuts there, I think some coffee, some smaller shrubs, and some ground cover, maybe even some vegetables. And we're getting a lot of food out of that system, but we're also providing those ecosystem services. This is good habitat, it's storing carbon, it's protecting soil. In Oklahoma, we don't have rainforests, obviously. We have a lot of tall grass prairies and other systems. So this is the Joseph Williams Prairie Preserve in Oklahoma. And it's also providing a lot of things we care about: pollinator habitat, bird habitat, soil cover, diversity. And when we convert those tall grass prairies to annual grains, we lose some of those benefits. The thing about the prairie that's different from the rainforest is that a lot of the benefits we care about are actually below ground. So prairie plants, prairie grasses, legumes, put most of their energy into their root systems, and they grow these deep, dense root systems that build soil health. And that's why some of our most productive soils around the world are prairie soils. A lot of our great farmland is across the Great Plains in places like Illinois, Iowa, throughout this whole region. And it's because those prairie plants, over hundreds of thousands of years, with their deep root systems, built these really deep, fertile soils. And again, as we replace these perennials with annuals like wheat, they have a much shallower root system, and that can lead to some problems with soil erosion. So one thing that makes it challenging to do natural systems agriculture in a prairie ecosystem is that the grains we grow across the world are annuals, right? And annuals behave very differently from perennials. So this group called the Land Institute in Salina, Kansas, started a plant breeding program to breed perennial varieties of grain crops with these 6 goals. So everything from soil conservation to economic stability for farmers. I did an internship here back in 2019, and that's where I first got introduced to this idea. So they're working on a few different perennials. This is perennial rice. It is grown in some parts of Asia. It's a cross between annual rice and a perennial rice relative. This is a perennial legume. It's actually sainfoin that's being bred for bigger seeds that you could eat like a lentil. And then this is kernza, which we're going to talk about, which is the one that is now grown commercially in the US. So again, here's a representation of that idea. Even though so on the right is a perennial grain, on the left is an annual, even though they have similar amounts of above ground biomass, because that perennial is growing for many years and putting more energy into its root system, it can grow these deep, dense roots, which means not only is it building soil health, but it can access nutrients and water down there that the wheat might not be able to. And here's what that looks like in real life. Here's this really deep root system of kernza and this relatively shallow root system of wheat. Not only is this good for stabilizing soil and building soil health, it's also great for the soil microbes. We know a lot of microbes actually require plant roots to live. They're symbiotic with those roots. Thanks. So when you have an annual system and the soil's bare for part of the year, there's no living roots, a lot of those symbiotic microbes die off. But in a perennial system, even when the above ground plant goes dormant, those roots below ground are still alive, they're still there, and those microbes can build up over time. And we'll talk more about those rhizosphere microbes later. All right, so backing up a little, what the heck is kernza? So kernza is intermediate wheatgrass, that's the species. It's a cool season sod-forming perennial grass native to Eurasia. It's planted widely across the Western US, especially where I am in Wyoming, as a forage grass. So it's a great forage grass for cattle. Have many of you planted intermediate wheatgrass or seen it or worked with it at all? I'm guessing it's a little farther north than here. Cool. All right, so this is wild type intermediate wheatgrass. There we go. This is wild type intermediate wheatgrass, has a pretty small seed, a normal grass seed. After about 30 years of breeding, this is what kernza looks like. So this is a variety of intermediate wheatgrass. And as you can see, those seeds are a lot bigger and plumper than the wild type, but they've still got a long way to go before they start to look something like a wheat seed. And that makes sense. Kernza's been bred for 30 years, and wheat has been bred, as far as we know, since prehistorical times, about 10,000 years. So this is a lot of progress in a short amount of time. Kernza yields are still low, about a third the yield of wheat at best, but yield is increasing year after year. In the past 15 years, that grain yield has about doubled. The current focus of the breeding program, there's a few now, one at the Land Institute, one in Minnesota, might even be a third. And those breeding programs focus on seed count and seed size, so yield characteristics, and also free threshing. Right now, kernza, it comes in a hull similar to some other ancient grains like spelt, and it requires a deholing step before it can be milled. But newer varieties are more and more, I think the newest variety in the research plots is 80% free threshing. So that is something they're making quite quick progress on. And here's a seed head for intermediate wheatgrass Kernza. All right, so what can you do with Kernza? A lot of things. The marketing team at the Land Institute has worked with a lot of different sustainability brands to try to include Kernza in some products. So Patagonia Provisions makes a Kernza beer. Kodiak Cakes makes these Kernza pancakes. It can be used a lot, very similarly to how wheat is used. Flours, cereals. This is my favorite. This rolled Kernza is kind of like a rolled oat. And more it is showing up. I actually saw Kernza beer in the Laramie liquor store just last week, which is the first time I'd seen it in Laramie, which is pretty cool. There's also some local breweries and bakeries that are experimenting with Kernza. It has a nice flavor. And so there are some local partners who are excited about using it. That being said, it's an experimental crop. It's extremely new. It's extremely small scale. There are 75 growers across the whole US who are growing Kernza in 15 states on only 3,200 acres. So just tiny acreage. 4 states across the Great Plains have planted Kernza in the last few years. As far as I know, it has not been planted commercially in Oklahoma, though Danny has some trials. But it has been planted in some neighboring states. And Kernza, currently about half of it is managed conventionally, and the other half is organic or regenerative organic, which is an additional certification. When we look at the markets on the next slide, I'll talk a little bit about why that is. And like I said, yields are really low, about 310 pounds an acre. That I think was the nationwide average for 2024, a little bit higher in the Great Plains, 380 pounds an acre. After the grain gets cleaned and dehulled, that's an extra step, and there is about 40% loss. So the final yield is lower. So that's kind of the negative, these really low yields. But one positive is that Kernza is multi-use. Most farmers who grow Kernza are not using it only for the grain. It's still a pretty high quality forage, and also can produce a straw. And because it's a perennial, you can get several harvests off of it in a year. So often farmers are taking a grain harvest off of it, and then that straw is still a lot higher quality than wheat straw, so it can be used as a forage. And then some farmers are also grazing it, maybe in the early spring or fall. Yeah, so there are some farmers who are using it for all 3 uses in a year. That's probably in a wetter environment. In Wyoming, we're seeing a grain or a hay harvest, but probably not both. We're very dry. What else did I want to say about it? Oh, stand age, Kernza lasts about 3 to 4 years before grain yields start to decline and it needs to be replanted. Grazing a Kernza stand does increase the life of that stand. It helps prevent it from growing into itself since it is sod forming, and keeps that stand kind of acting young for a few extra years. There are also farmers who will get 3-ish years of grain harvest off their Kernza stand and then move into a few additional years of a hay crop before replanting that stand or rotating it into something else. Okay, marketing Kernza. This isn't my area of expertise, but the people at the Land Institute sent me some information about this because I know it's a big challenge right now with Kernza with such a new crop. Here are some of the big market challenges. So things like processing, you need a place to process that smaller seed. There are processing facilities in Kansas. Aggregation and storage for such a niche crop is hard. And then consumer awareness and demand and matching up that supply and demand with such a new crop are all challenges. The price is pretty high. Right now it's selling for about $2 a pound conventional and more than double that organic, which is more than 20 times the price of wheat. But the conventional market, at least if you're trying to sell to some bigger buyers, is struggling right now. There's conventional grain in storage. The organic and regenerative organic markets are much stronger. And again, that's because a lot of that grain is moving through these sustainability brands like Cascadian Farms and Patagonia Provisions that want that organic certified grain. The farmers I worked with in Wyoming, we'll talk about this a little later, were able to get their grain sold if they were able to find local partners, brewers or bakers who wanted it, but it can be harder to market, especially conventional grain. Yeah, just on broader markets. Okay, so now we're moving kind of into specifics. I'm going to talk about the Kernza work I did in Wyoming. One thing I wanted to say here, actually, that price is really high and the yield is really low. The expectation is that as supply increases and as yields start to go up, that price will start to come down. So that's what we're seeing slowly over time. So part of my PhD work, I worked with 4 farmers to plant Kernza for the first time in Wyoming. And we just wanted to see, can Kernza even be grown in that climate? And how does Kernza impact soil health compared to wheat fallow or a perennial grassland? So this is the ecosystem of Wyoming that I was working in. This is southeastern Wyoming, near the Colorado and Nebraska borders. And as you move across the Great Plains, it gets drier and drier, as you all know. And we actually get about 100 miles of dryland farming in southeastern Wyoming before it gets too dry to grow dryland crops. So this is really the limit of dryland farming. Not only do we have really low precipitation, so about 15 inches a year, it's really variable. Since 2008, we've seen anywhere from 6 inches in a year to 19 inches in a year. So that variability makes it hard to be a farmer, of course. And it also makes this land really vulnerable to things like erosion, land degradation, and climate change as drought is changing. All right, so I wanted to look a little bit into the Oklahoma climate and some trends. So I work on climate resilience and drought adaptation in Wyoming. These climate quick reference guides, I really like. They're created by the USDA. They're available for every state and also for every specific county across the US. So I highly encourage anybody to look up the reference guide for their county. And what we've seen across Oklahoma is an increase in precipitation of about 25%. And so far, only slight warming, about 0.5 degrees, and most of that warming in the winter and the spring. That being said, these guides also show you the top causes of crop loss, insured crop loss, which is drought, like way more than anything else for Wyoming. And even though we're seeing that increase in precipitation, the expectation is that most of that increased precip is happening in the winter, fall, and spring. And Oklahoma is actually expected to have drier summers. So a big decrease in precip in summer and an increase in other seasons, which means we expect to see more summer drought. All right, moving back to Wyoming. This is a study that really shows a lot of these principles that I was interested in with Kernza. So this study happened in that region of southwestern Wyoming. And it shows that some of those principles of natural systems agriculture. So this is our native system. It's a short grass prairie / sagebrush steppe. And as that native system was converted to wheat fallow, this historic system, we lost over half of that soil organic matter. That bar went way down. This historic system includes 14 months of fallow in between crops to store water. And during those 14 months, the soil is bare. There's often heavy tillage to keep weeds out of the fallow. And we have a lot of wind. So sometimes that soil is blowing around. And that caused a lot of soil degradation and started to decrease crop yields in this region. As that system was converted to a minimum till and a no-till, a lot of that soil carbon was able to recover. And if these systems are planted to perennial grasses as part of the Conservation Reserve Program, the systems recovered 90% of their soil organic carbon in just 15 years. So soil health is really responsive to change, even in these dry systems. And one thing to note is that these are not native grasses. We're not perfectly mimicking this sagebrush steppe. These are introduced forage species and some alfalfa often. But because they're perennial and they act very similarly to these native perennial grasses, they're really doing a great job of improving soil health. So like I said, we worked on 4 farms, but the data I'm going to show you today is from one farm. It's from this farm in Chugwater, Wyoming. And this farm used to be all wheat fallow at some point. About 10 years ago, this big wheat fallow field was planted to CRP, a mixture of perennial grasses and legumes. And then at the start of this project in 2021, this field was planted to Kernza. And then we sampled the soil. Sorry, these titles are messed up. 1 year after Kernza planting or 2 years and 3 years. And we sampled the soil at these 2 depths. Yeah, and the reason we sampled these 3 systems is that we wanted to use the CRP as our positive control. We know soil health is improving in that system. And the wheat fallow is our negative control. We know that fallow is not great for soil. All right, so thankfully Danny explained all of these soil health analyses, so I'm not going to go into this too much, but we measured all sorts of things, measurements of soil structure, chemistry, and then organic matter pools, ranging from these stable pools to these more labile pools that cycle more quickly, as Danny was talking about. We also measured soil microbiology. I'm going to kind of skim through this part, but basically we looked at microbial activity, biomass, and also did a little DNA work. The first thing we found was no change in the bulk soil organic matter under Kernza. This kind of makes sense. These systems are really dry, and after 2.5 years, that's probably just too short to really see a change in organic matter. So yeah, not much change there. The exception was mycorrhizal fungi. In year 1, not much difference between the Kernza and the wheat field, but in year 2, the Kernza had way higher mycorrhizal fungi than the fallow. So that makes sense, right? Year 2, that field was fallow. There were no living roots. Mycorrhizal fungi need living roots to survive. So the Kernza field had a lot more of those fungi. All right. Another thing we did was look at those roots. We know that when we think about perennials, when we think about prairies, we got to be looking at the roots. So we got a bunch of people together and we went to these fields and we dug up a whole bunch of plots across the fields. And we took these roots, we rubbed all the soil that we could off of them into these buckets. That was the rhizosphere soil we collected. And then we took the rest of the roots home, washed them, dried them, and weighed them. And this is what we found. So in our really dry system, Kernza did not produce this 3-meter deep root system. That makes sense. A lot of our native grasses don't do that either. But it did produce about 3 times as much biomass, root biomass, as those wheat plants. So here's that root system of wheat, and here's our root system of Kernza. It is much bigger. And it even produced more root biomass than the CRP, likely because those CRP plants were just really old at this point. And there was a lot more dead and dying root biomass around there. So this is a great finding, and it shows us that soil health is likely starting to improve because we know how important root biomass is for the soil. So we also collected that rhizosphere soil. So the rhizosphere is that zone of soil that's intimately connected to those plant roots. So it's a soil that's attached to those roots. And in the rhizosphere, there can be 100 times as many microbes in that rhizosphere soil as in the other soil. And that's because plants exude a ton of their energy, about 1/3 of their energy, into those roots to feed those soil microorganisms. Why do they do that? Why are they giving up so much of their energy? That's because those microbes are helping plants. They're delivering nutrients to those plants. They're keeping them healthy, preventing disease, and also helping with stress responses. A lot of people think of the root microbiome the same way we think of our gut microbiome, doing a lot of those good things for us. Here's pictures of root tips, and as you can see, they're just totally covered in microbes. This is a picture of a plant seedling. All of these darker, thicker white roots are its own roots. And all of this fuzzy stuff is that mycorrhizal fungus, which is one of the main symbiotic microbes we like to talk about. And so most of the soil, nutrients, water that this plant can access is not even through its own roots, it's actually through that mycorrhizal fungus. I like to bring up this quote: "Roots evolved in a microbial world, and ecosystem functions and services for roots cannot be considered in isolation from these microorganisms." All right, so what happened when we looked at that rhizosphere soil? What was the difference between wheat, these CRP grasses, and Kernza? Well, what we found is that Kernza looked a lot more similar to these perennial grasses in most of our indicators, so these organic matter pools, than wheat. So that meant that the Kernza is exuding a lot more organic matter through its roots than the wheat is. When we looked at the microbial community, we also found something pretty interesting. So these graphs kind of represent the composition of those microbes, and how you read them is the more the circles overlap, the more similar those microbial communities are. So for bacteria, each of the 3 crops had pretty different microbial communities. But for mycorrhizal fungi, there was a lot of overlap between the Kernza and those CRP grasses. What does this really mean functionally? What do those different species of mycorrhizal fungi do? We don't quite know yet. So this is really an exploratory finding. But another interesting thing is that, we think of, the mycorrhizal fungi, maybe you've looked at inoculants, they have a few types on there, but we found over 4,000 different types of mycorrhizal fungi in just the rhizosphere of these 3 plants. So there are so many different species and types of fungi that are important to these plants, way more than just maybe if we think about a couple. All right, so we're moving on from the microbial stuff, and we're going to get into what you guys maybe care more about, which is the agronomics. So first thing I want to say is that in the 2 years we collected yield, we had very different environments. The first year was a drought year. We had 20% below average precipitation, a really dry spring and summer. The second year was a wet year, 40% more precipitation than average. So that's really driving these findings. First thing we measured was above ground biomass, because Kernza and intermediate wheatgrass is a really important forage crop. And we saw that Kernza produced pretty good forage even during a drought. So again, not as much above ground biomass as wheat, but it is a higher quality forage than wheat straw. What we saw for the grain was different. So in our drought year, even though Kernza was able to survive that drought and produce some good forage, it produced almost no seed yield, really not even enough to harvest, like maybe a few bushels an acre. In a wetter year, we still had low grain yields, about 5 bushels an acre, but we did produce enough to harvest. And that's compared to about a 30 bushel an acre yield for wheat, which is pretty typical for our region. We also had some irrigated Kernza as a comparison, and the irrigated Kernza was able to produce about 10 to 20 bushels an acre. So it really was water limited in our environment. So again, these yields look so low, but the Kernza price is about still 20 times that of wheat. And just to put this in comparison with yields across the US, because this is data from only a few farms, a few years, it's so preliminary. But if we think about those average yields across the Great Plains and across the US, 5 bushels an acre is 300 pounds, and that Great Plains average was 380 pounds. So we were not too far off. All right, so the real reason we're interested in Kernza in this environment is not to get huge grain yields. We know that's not going to happen, but we're really interested in Kernza because of its flexibility. So in this region of southeastern Wyoming, we're seeing that declining soil health, worsening drought, and also poor markets for crops like wheat has led to a lot of cropland abandonment. There are a lot of farmers who are saying, this isn't penciling out anymore. I'm moving into maybe a grazing system or planting everything to CRP or selling my land. So the idea is that maybe Kernza could provide some flexibility for those farmers who are really looking for other options. And that's because in a dry year, Kernza can be used as a forage, right? And it's a pretty good forage crop. A lot of farmers plant intermediate wheatgrass as forage anyway. And you don't necessarily see crop failure in the same way if you plant wheat and you have a drought, you're really in the hole because Kernza will survive that drought and come back. And if you get a wet year, you can still get a grain harvest off of that crop. All right. We did an economic analysis. I'm not an economist, but I'm putting this all up in case people have questions or if you want to look at it later. But some basics from that economic analysis. So we worked with an economist. For Kernza, we assumed that in our environment, you'd get a grain yield in wetter than average years and a hay yield only in drier than average years or wetter. Yeah. Anyway, so every other year you're getting grain, every other year you're getting hay. Whereas for a wheat fallow system, every other year you're getting a wheat yield, and then you have a whole year of fallow in between. We assumed 5 bushels an acre, which is what we found, compared to 30 bushels an acre for wheat. A much higher price for Kernza, $2 a pound. And some other differences in those big spreadsheets we looked at was that Kernza has lower input costs. It does not need to be planted every year. It's more competitive with weeds since it is a perennial, so less weed control costs. But it does have that deholing cost, that processing step that adds some cost. Whereas for wheat, you have higher machinery and planting costs because you need to do things like till and plant every year. So once we run all those numbers, and that includes everything, that includes labor costs for running the equipment, some opportunity costs, we found that we get a small positive net return for Kernza, $11 an acre, and a negative return for wheat fallow. And that does kind of make sense for our region. Like I said, we're hearing a lot of farmers say, especially these last few years, this has not been working out, and we're looking for alternatives. All right, so that data was so preliminary, right? First time it had ever been planted in Wyoming, only worked with a few farmers. So we wanted to run some hypotheticals to ask the question, what kind of scenarios need to exist for Kernza to work out? So what we did is we reran this analysis and we looked at, these are net returns for just 1 year of grain production. So we're not considering the hay year, we're not considering any fallowing, just looking at 1 year of grain. We saw that, and we used the price and the yield from that last slide as our average, and then ran a few hypothetical higher, lower yields, higher, lower prices. And we found that under most scenarios, Kernza will produce a positive return if you get a grain yield, unless you're getting a low price and a lower average yield. So this is just kind of a thought experiment, but this tool is available, this spreadsheet is available online, and you can go in and plug in your own numbers and start to think about, okay, what is it costing me to run my combine? What kind of yield do I think I'll expect on my farm? And you can see if it might pencil out for your region. Yeah, so that was it's a little confusing. That was before it gets deholed. So you lose like 30, 40% when it gets deholed. And so this is a little lower than that. I don't know. It's standard if you've ever worked with another crop that needs to be deholed. Like if there's somebody who's deholed like spelt, or I'm not even totally sure the other ones. Yeah, it'd be similar. I know when I've talked to farmers, the main thing that has been hard has been shipping costs, because where we were working, there was not a deholer close by. The shipping, I think, was more than the deholing cost. But there is a Kernza specific processor in Salina, Kansas, which is closer to you guys. All right, so some conclusions for soil health. So that higher root biomass and more organic matter that Kernza is putting into the soil makes us think it's pretty likely that soil health is starting to improve in a Kernza system. And also, despite those lower yields, Kernza's higher price and that added flexibility could still make it a good option for some farmers in risky environments. I want to showcase a few resources. I know this was like, I barely touched on a lot of the stuff that I could have talked about. But if you're interested, this is a extension bulletin through the University of Wyoming that has that budget I was talking about, and it has a link to that Excel sheet that you can that you can play around with. And you can find that on the university website. Hannah Fancher at the Land Institute works with growers. And so if anybody is interested in thinking about growing Kernza or talking to somebody more about it, she'd be a good contact. And then this Kernza grower guide has a lot of those specifics about things like planting density, planting dates, weed management. So feel free to take a picture of this slide or come up and tell me, and I can send you some of this information. And I think they'll be posted as well. All right, and with that.
Oh, no, bad picture.
Yeah, any questions? I think we have,
I have a question back here.
Actually, I have two of them.
Yeah.
How long does the seed last in the soil, and how do I kill it? Because you're technically introducing a new weed down here in wheat country.
Yeah.
And, you know, the cattle producers, 40 years ago, we loved Italian ryegrass because of the forage.
Yeah.
Now we got an Italian ryegrass problem.
Yeah.
So how are we going to keep this from becoming another Italian ryegrass problem in our wheat fields?
Yeah, that's a great question. And the answer that I've heard to that is that, well, because intermediate wheatgrass has already been planted for so long across the Western US, that question has been answered a little bit, and it has not become a big weed problem in that region, intermediate wheatgrass. And as it's been bred into Kernza, usually when you breed a forage to put more energy into seed yield, it usually becomes less competitive. So you can kill it through tillage and with herbicides. Both of those are used. Yeah, and it hasn't, I mean, there's always the chance, but at least in that region, it hasn't seemed to have a lot of problems spreading beyond where you've planted it.
But normally those wheatgrass fields that got planted are still in wheatgrass after 25 years later.
Well,
Yep.
I do work with some farmers who do a rotation between forages and a annual grain, and they've had good success either tilling it out or spraying a herbicide,
Thank you.
at least in that environment.
Yeah, to be fair too, I mean it's a perennial, so it's not like super competitive on an annual basis as well. Yeah, true. It's changing. Yeah.
Since it promotes like a long-term root system, do you have any evidence about like another pathogens in the root?
Ooh, I don't know anything about pathogens in the roots.
I know that about like fusarium is a pathogen for root, for example, so I would like to know if it's.
So I have heard some about fusarium and ergot in the seeds, similar to wheat. Because Wyoming's so dry, we don't tend to have a ton of pest problems or disease problems. But yeah, if you wanted to know more, I could connect you with the Land Institute disease ecologist. But that's kind of all I know.
So is harvesting done with normal combine like wheat? I mean, does it dry down like wheat?
Yeah, harvesting's done with a normal combine. It is tricky. So thankfully, the farmers I worked with had experience harvesting grass seed for like a cover crop or for CRP, and setting their combine for that. But I have heard people having trouble with not setting their combine right and just driving right in and blowing all their seed out the back. So you do have to be careful about how you set your combine. And there are some people you can contact, I believe at the Land Institute, who can help walk you through that. But yeah, it's just tricky because it's a grass seed. It needs to be planted a lot shallower, and the combine settings need to be different for that small seed.
Maybe one more. If you did let it go to seed on its own, say like in a marginal year, would it reseed itself just by its seed drop instead of having to replant it?
Would it reseed itself? I don't know. It mostly reproduces in the field rhizominously, so it's going to spread and send up more tillers. I know in that CRP field there was intermediate wheatgrass, and it was still there after 10 years, but it was hard to tell how much of that had been reseeded and how much of that was just perennial and had stuck around for that long. Yeah. All right. Thanks.
Thank you, Hannah. All right, to round out this session, Dr. Galvin's going to talk again about some of the ecology-based stuff she's working on for weed seed bank management, and hopefully I have an opportunity to follow up and see what that does for soil health eventually. So thank you, Liberty.
Hello. Okay. Great talk, Hannah. I'm glad somebody asked the weed science question in the back, because when you put up that picture, I jumped. I was like, oh my God, it's an Italian ryegrass plant. What are we doing? No, but the perennial piece, I think, really helps. So, last but not least, right before lunch, I'm going to do my best. We're going to stay on time. So last year, many of y'all have probably seen me at a continuing education event. I get a lot of people that come up to me and say, Liberty, I can't get my herbicide resistant weeds under control, and I'm going to go back to diesel and steel. And these are people that have practiced no-till for a really long time. And Jason Warren was my soil and water conservation teacher. So when I hear people say they're going to go back to diesel and steel, it upsets the little soil scientists in my heart. So I was talking to Dr. Adamson, and I was like, hey, we should like look into this. You know, what are the trade-offs? I know a lot about seeds. So I'm going to talk to you a little bit about the literature and then some preliminary data that we have. So I'll talk about seed bank dynamics, the intersection of weed seed bank management with soil conservation, and then the current research that we're doing. So this is a really good book, No-Till Farming Systems for Sustainable Agriculture. Jason Warren probably wrote this book. Just kidding. He's not a co-author, but I think he used this book whenever I was in class. But in the beginning of this book, one of the major issues that they talk about for long-term no-till farming is herbicide-resistant weeds. And because I work in wheat, I mostly focus on those annual grasses. In no-till, they're all falling to the soil surface. But if they're resistant to imazamox, quizalofop, and glyphosate, what are we going to do about it? Okay. So before we get into it, weed seed banks are dynamic systems. They're influenced by the weather, moisture, etc. But I like to think of it as like an actual bank. Okay, so you have your withdrawals and you have your deposits. Unlike your own bank account, you don't want to make deposits. Okay, so a deposit can happen when a plant goes to seed and it drops all that seed in the field. Weed seeds can also make their way to your field with machinery. I get a lot of people that love to blame custom cutters who don't clean their equipment for all of the weeds that they have in their field. That's none of my business. Animals, a lot of the annual grasses that we have can pass through the gut of a ruminant. And so people that have weedy grasses in their pasture, maybe you don't consider it a weedy grass because it's a forage, but the dual purpose wheat producers that I work with, everything looks like a forage in the fall, and then they're pissed off in the spring because now all that stuff looks like a weed. And those can move field to field in grazing animals. Now, seed dormancy is its own thing. Seed dormancy is somewhat complicated. So there's a couple of different ways to define dormancy, but as soon as that seed falls off the plant, that's considered primary dormancy. That's sort of a dormancy that's naturally built into that plant. There can be different types of dormancy. So there's a physiological, what are we calling it? A mechanism, coat-imposed dormancy. So those are seeds that have a really, really hard seed coat and require scarification or some kind of like physical degradation before they germinate. The seed itself on the inside of that seed coat is ready to germinate. What happens is that physical seed coat is so hard that it will hang out in the soil until it's been beaten to death, and then that seed's ready to germinate. So that's the mechanistic. But then there's also embryo dormancy. So that's the seed itself has a sense. I'm not going to talk about CO2 respiration and things like that, but the seed itself has a mechanism to sense when the environmental conditions are right for germination and growth. So one is a physical barrier, the other is a biochemical barrier. So very generally speaking, annual weedy grasses, like the nasty ones that we deal with in wheat, our brome species, jointed goat grass, Italian ryegrass, they only really have a 3-year dormancy. Like longevity in the soil is not the way that it's not their life history strategy. And a weedy grass, if you think about the coleoptile of a wheat plant, if your soil is really tough, that coleoptile is going to have trouble moving through the soil. Okay, so with annual weedy grasses, they don't have a long dormancy. They also can't germinate very well from depths deeper than 1 inch in the soil. Okay, so with grasses, it's about 3 years. Broadleaf weeds, it could be lifetimes. Okay, do I have time? Yeah, okay. I got into a fight with a friend of mine. She's an astrobiologist. I don't care about space. She really cares about space. And we were joking about time. She was like, time is an emergent property of the universe. And I was like, time is relative and it doesn't matter, whatever. And so we started this argument about like, what in our world does not care about time? And if anybody has seen any of those studies from I cannot remember the name of the place, but there's a place in the UK where they discovered a bunch of seeds in jars. And these seeds were like 300 years old. And they pulled the seeds out of the jar and gave them moisture and the right temperature and they germinated. Okay. So in my opinion, weed seeds they're not beholden to time. Emergent property, blah, blah, blah. Seeds will last as long as they can, depending on the environment that they are in. Okay. So they also have very different dormancy periods. So what we're looking at here is There was 7 different states that participated in this study, and they basically all buried weed seeds at different depths. And this was in like the north central. So I think it was Minnesota, Colorado, Michigan, Wisconsin. They get really, really cold winters, like colder than the winters that we have. And every year they measured emergence from these, they measured germination from these seed packets. And you'll see the grasses, that's 31% in the first year. This is kind of related to why we say the dormancy period is about 3 years, because all those seeds fall to the ground in the first year. 30% are going to germinate and emerge. In theory, if you don't add any more seeds to the soil seed bank, in that second year, another 33% are going to germinate and emerge. And then in the third year, in theory, you're going to be down to a very, very small seed bank. That is not the case with our broadleaf weeds. And I want to point this out. I don't like this. I read a different paper that had a much lower number than this. But then I've seen other research, like, I don't know if he's in here, but I think Maxwell Smith and I were talking about some pigweeds have like a short dormancy, and then other varieties have a longer dormancy. So it depends on the species that you're dealing with your amaranths, but they have a much longer dormancy lifespan in the soil. So the withdrawals, we are living life in the soil. We are withdrawing all the freaking seeds that we can. Okay. Now a withdrawal is germination. And I think as agronomists, like, we don't want to see weeds in the field, but from a seed bank management perspective, the more seeds that we can germinate and get out of that seed bank, the greater the opportunity it is for us to kill them in whatever fashion you want to kill weeds in. There's also some really interesting soil pieces about decay. So somebody asked me the other day, they were like, how does glyphosate influence the soil microbes? And actually, soil microbial degradation is the way that glyphosate breaks down. Glyphosate does not have a residual in soil, and that glyphosate presence, there's some studies that say it actually increases the microbial population because you're adding food to the soil. Okay. Similarly with your weed seeds, if you have a diversity of weed seeds in your seed bank, that sometimes looks like a buffet to these pathogens. Okay. There's some really interesting research I didn't put into this talk about crop rotation. So when you rotate crops, you're also kind of encouraging a diversity of weed species depending on the crop that you plant. And then you get a diversity of weed species in your seed bank, and the pathogens love that. Okay. And then predation. There's little bugs, little creatures that like to eat seeds. So if we stimulate germination, we have an opportunity, but if you're a no-till producer, like, what's the risk? Okay. And that's really what I wanted to get to today. Just walk right behind me during my presentation, Desiree. This is my student. I have to point them out because you came in halfway during my talk. That's fine. Okay. So this paper is from 1992, but it's very fascinating because they look at the weed seed bank responses to tillage, herbicides, and crop rotation. I'm not going to focus too much on the crop rotation part, but if y'all are interested in that, the data was super fascinating, and they're actually finding a decrease in the weed seed bank with more crop rotation. But after primary tillage, this is how the weed seeds shift in the soil. So we have inches on the left side, 0, 2, 4, 6 inches in the soil. When you use a plow, 53% of whatever's sitting on the soil surface is going to fall down to the depths. Okay, when you use a chisel, you're not going to get the same amount of distribution through the profile. Now, this was just one pass, take this with a grain of salt. But to me, this is fascinating because I know that those winter annual weeds and the grassy weeds that we work with, because of that coleoptile factor, they can't germinate from depths that are, their optimal germination depth is a 1/2 inch. Their optimal burial depth is a 1/2 inch. And you will see an exponential decrease in emergence as your burial depth gets deeper. Okay. So to me, I was like, all right, yep, we don't even need to do this study because if we bury everything at 6 inches, we're good to go. That's not the conclusion we came to. Okay. So then what are the influence? So what actually happens in the soil when you've been doing no-till for 20 years and then you decide, I have a weed problem, I'm going to bury them. I'm willing to take the risk and see what's at 6 inches and bring that up to the soil surface. I have an undergrad who we're going to actually do this tillage experiment next summer. And he is 24 and he said his dad put away the plow when he was born. So the property that he farms, he's got 24-year-old no-till fields. He's got a windmill tumblegrass problem that he cannot manage. And he and I have talked about it a little bit. And he was like, I want to try some tillage. I want to do like a little tillage thing. And I said, well, don't go back to conventional tillage. Like, let's figure out what an optimal tillage application looks like and then monitor the seed bank to see like what's the outcome. Okay. So in this particular paper, they found a lot of benefits. My favorite one, it says controls herbicide resistant weeds. Seems like a bold claim. They didn't really provide a lot of evidence for this, but okay. So here's the highlights from that particular paper. Yeah. Okay. So occasional tillage does reduce carbon, right? And we're very concerned about that in this environment. But it has limited effects on the soil properties and crop yields. I think for this study, they had 10-year-old no-till fields and they did tillage at 5 years. Okay, so they only did it once, which is also what I'm kind of proposing. And the beneficial occasional tillage depends on the timing, the depth, right? Same factors with any other agronomic practice. The metrics of that particular application are what's going to define it as successful or not successful. And my favorite, did you read this one, Brian? Occasional tillage once in 5 to 10 years may not undo no-till benefits if erosion is controlled. And that's something that Dr. Arnell and I have talked about. Like, okay, if we till the soil, how are we going to keep the soil from exiting the field shortly after tillage? Because it's been under no-till for 20 years. So this study, they looked at, it's a global meta-analysis. So they didn't necessarily do the research themselves. They took everybody's research who has done any kind of tillage and no-till research. And I really like the graphics that they came up with. So over on the left-hand side, it says conservation agriculture. Let me use my pointer. Conservation agriculture, when exposed to occasional tillage, has positive impact on weed control. So this is a plow, this is a chisel, this is a subsoiler. Has positive impact on weed control, relatively neutral impact on chemical properties, positive impact on physical properties, neutral or positive impacts on biological properties. The plow is going to negatively influence your soil organic carbon. Neutral impact from these other chisel and the subsoiler. I don't know why these are blank, but positive impact on soil erosion, which is an interesting claim. But no change in crop yield with the plow or the chisel, and positive change from a subsoiler, which, okay. This is, so, and keep in mind, a meta-analysis is like hundreds of papers that they have sort of compiled. So it's not necessarily our environment that we would get all of these pluses or minus, but generally across the world, these are the trends that we're seeing. Okay, and then I really, really like this graphic. So this is also from that same meta-analysis paper. So factors influencing crop yield are up here: climate, agronomy, soil properties. And with occasional tillage, they are finding that maize yield across the globe went up by 4.4%, wheat yield went up by 6.3%, rice yield up 2%, and legume yield up 1.9%. The blue arrows are sort of a negative. You can think of that as like a negative outcome, and the red arrows are a positive outcome. So increase in yield is a positive outcome. They found bulk density decreased a little bit. The soil water content improved. Soil porosity improved. Total nitrogen at this depth, so 0 to 20 meters, that's 8 inches, 0 to 8 inches. Total nitrogen decreased. Soil available phosphorus in these upper depths improved, but did not improve at these lower depths. pH improved in the middle of the profile, but did not improve at the lower depths. And then soil organic carbon at the surface did not improve, but soil organic carbon at depth improved by 5.1%. So these are definitely the trade-offs. And I know that, if you participate in the carbon credit program, maybe this is not attractive to you because soil organic carbon is the thing that takes the biggest hit, but there's a lot of other things involved that are not going to suffer a detriment. Okay. So the study that Dr. Adamson and I started, if you went to the field day in Chickasha in the spring, that's what I talked about there. That's not a no-till field over there. We just were really trying to figure out our methodology for soil sampling, But what we were finding is before we did our tillage events, there were seeds that were buried deeper than 2 inches that were still viable. These were mostly broadleaf weeds. So here's actually a picture of some seeds. Anybody know what these are? Anybody want to guess? Just do your best. This one is probably not a weed. It's okay. So this is actually proving my point. It's really hard to identify seeds, right? So if we're doing this seed bank analysis, seed identification has been a big companion project to this particular project because we want to know what we're pulling up from the depths. But post-tillage, weed seeds on the surface decayed when they were buried at 2 inches. So, not sure what was happening there. I was talking to my technician about it. I was like, where did they go? Like, why aren't we seeing these seeds? They were there at the top. We just buried them. Not a big deal. And he said, I don't know what happened. So maybe we screwed up our sampling process. Maybe they did decay and die. We also ran a tetrazolium test, which is basically just a colorimetric test. You slice the seeds open and you put a little tetrazolium on them. If the tetrazolium turns pink, they're viable. The majority of the seeds that we found were viable. And when we did this 2-inch sampling, this one, they were also they had decayed a little bit. So that tetrazolium test was not pink. So that was really fascinating. I have engaged my undergrad. He would really like to do this project on his property. He has jointed goat grass, windmill tumblegrass, the occasional bromus species. He does not have Italian ryegrass. But we are potentially looking for other collaborators that are interested in this research. And we have gotten our methodology down to a T. Connor is really, really good at finding needles in a haystack. That's his whole PhD. So the other thing too is like, well, if we're doing tillage, we're not just doing tillage to bury seeds. Like, let's do tillage as a weed control measure. And what we found is that, as you would probably expect, like a shallow disc, we only really got 40% weed control, like visual weed control at the soil surface. And then our moldboard plow plus a disc afterwards to kind of level everything out. We got 95% weed control. So it's kind of a duality. Like, if you're going to till because you're trying to get rid of your weeds, till as a weed control measure as well. Like, do it at a time when you're going to also kill some plants. Okay. So, I nailed the timing. Okay. So that's it. That study is going to get kicked off next summer. Dr. Adamson and I applied to a Southern Sayer grant and we were rejected, but they really asked that we resubmit that proposal. There's $25,000 worth of farmer payments as part of that grant. So if you're interested in getting paid, call me. Okay. And there's actually a lot of USDA grants and other regional grants that have started to really ask for farmer involvement, producer involvement, and they involve money. Okay. So if you're interested in something cool, you want to get paid for it, would be happy to talk to you about collaboration. Okay. Yep. I can take questions if there's questions, sir.
Could you provide more context on the slide where we saw a decrease in bulk density, which I'm assuming means, yeah, it's getting more compact, but we're seeing an increase in soil porosity and an increase in soil water content. Aren't those vice versa?
So this was like a global study, and it's not one individual site that experienced like one thing or another. So I agree, these are a little bit contradictory. I actually should look at that paper. He's shaking his.
It's actually
But
a good thing. This should be a positive. This should be a positive.
Okay. I think the paper said that any blue arrow was a negative impact.
Positive. You don't want him. Or I mean,
I still think
bulk density and increasing bulk density.
hello. But the point I'm trying to make is I think in this paper, the way that they categorized the arrows was a negative impact and a positive impact. You're right. You're right. So I'm going to go with the boss over here and say it went down. Yeah. I would be happy to talk with you more. I can go and look at the paper and just see exactly like how they did their methodology, because they may elaborate on this a little bit. I agree. That is confusing. Yeah. Contradictory. Yeah.
Okay, I also have a question about this meta-analysis. Do you know how long after the occasional tillage they were looking? Like, were they looking at the impact of occasional tillage after every 5 years, after 30 years, or like a year after they did one occasional tillage event?
I need to stop turning this off. I need to look back at the paper, but they did. I think they did occasional tillage more than once, and they monitored it through time. But because it's a meta-analysis, there may have been like different sampling periods. So let me look at the paper and get back to you on that. Yeah, yeah, because the proposal is not conventional tillage. We're not going back to conventional, and both of these papers said one big ass tillage event every like 5 to 10 years, you shouldn't suffer. You know, assuming you don't get like a massive flood that now carries all of your soil into the creek, you know. Yes, sir.
Have you looked into on your seed survivability? Have you looked into difference in soil surveys and see if that changes, whether one survives 2 years or 3 years, or one degradates down in less than a year with the different soil types?
So I haven't looked at that, but that is definitely a factor. The anaerobicity. So if you have like a really, really heavy clay that has less infiltration, it's going to get less access to moisture, which will probably make it stay in the soil longer. All seeds need the right temperature and moisture to germinate. So if it's stuck in an anaerobic environment, it may very well stay dormant for a longer time, or it might decay. The plan is to find collaborators and just work with whatever soils we get. But the ideal scenario would be we find different soil pHs and different soil textures. Yeah, because those factors definitely influence dormancy and termination.